this recording, we will discuss the structure of skeletal muscles. Skeletal muscles fibers are formed by the fusion of many embryonic myoblasts. This is how we end up with skeletal muscle fibers that have multiple nuclei. Okay. The myofibrils within the myocyte or the muscle fiber cell, these are the most abundant organelles within these cells, and this is how we get the contraction. Remember, these myofibrils are really where your contractile proteins are. Okay. So if we go back to our familiar image, these large circles are the myofibrils. Okay, this is where your specialized contractile proteins live. Okay. The blue surrounding area, we mentioned previously and briefly, sarcoplasmic reticulum. Okay. Said we're not going to make proteins, we're not going to make lipids, we are actually going to store and release calcium ions. Your sarcoplasmic reticulum stores and releases calcium ions. Write it down, highlight it, put a star by it. Okay? Calcium is going to be a big deal. All right? So notice that those sarcoplasmic reticulums surround every single myofibril. Okay, so you've got calcium all around these contractile proteins. Write that down. That's important. All right, couple of more structures. Transverse tubules, or T-tubules, as we'll call them, are inward extensions of the sarcolemma that surround the myofibrils. And so we can see we are going to take the sarcolemma and we're going to stuff it down around the myofibrils. Okay, and we do it on both sides equally. Okay, so these little tan colored lines, okay, those are the T-tubules. Okay. These are going to create tunnel-like networks within the muscle fibers. The inside of these T-tubules is continuous with the exterior of the cell, which means these T-tubules are full of extracellular fluid. Okay. Write that down. Jot it down. Highlight it. Something that's important. Okay. That extracellular fluid is going to have some pretty good stuff floating around in there to help us with muscle contraction. Okay. You also have terminal cisterna, which are enlarged regions of your sarcoplasmic reticulum, and you find these on either side of your T-tubules. Okay, this whole composition of cisterna, T-tubule, cisterna, this all together, these three pieces, is a triad. Okay, so you've got triad after triad after triad. Okay, now we keep mentioning them. These myofibrils. Okay, we're going to go into a little bit more detail. You know, buckle up. Each of your myofibrils has hundreds, maybe thousands of myofilaments, okay, inside of them. These myofilaments have one or more of these three proteins. We have contractile proteins, regulatory proteins, and structural proteins. Your contractile proteins are in fact going to contract by generating tension. Your regulatory proteins are going to regulate or dictate when these fibers can contract. And your structural proteins are in fact going to provide structure to maintain proper alignment and stability of the other fibers. Okay, so everybody has their own role. There are three types of myofilaments that we're going to chat about over and over and over. We have thick filaments, thin filaments, and elastic filaments. Thick filaments really just come in one variety. These are contractile proteins known as myosin filaments. Okay, so myosin are your thick filaments. Thin filaments come in varieties. We have actin, tropomyosin, and troponin. Okay, so all three of these are thin filaments. And then we have elastic filaments. Okay. The one that we are going to chat about very briefly is titin. Okay. Titin is a giant spring-like structural protein that stabilizes the overall structure that we're getting ready to talk about, and it helps resist excessive stretching. Remember, one of the things that we would prefer not to do would be overstretch your muscles. Titin helps prevent that. So this is what it all looks like. So this upper part of the image should look familiar. Right. This middle part maybe look a little familiar. This is what we're going to focus on now. Okay, so muscle fibers composed of myofibrils. Myofibrils are composed of these three proteins, okay, these three filaments. Your thick filaments, your myosin filaments, okay, this is one individual myosin. They arrange themselves in these giant bundles. Your three your three thin filaments include actin. Okay, each one of these purple dots with the divot, okay, each one of these is an actin, so you can see we've got a particular arrangement here. We've got the troponin, which are these little yellow ones, and these blue strings are your tropomyosin. Okay, so all three of these are your thin filaments. And then this is titin, this is your elastic filament. Okay, and you can see how many of these we have as well. All right, let's talk about what everybody does. Your thick filaments. Okay, we said that's myosin and only myosin. We have a head, a neck, and a tail region. Okay. These tails are going to intertwine. Some heads are going to go one way, some heads are going to go the other way. Okay. We've got that hinge-like neck that allows us to bend. Okay. You may or may not have noticed, but this head shape, go back here, this head shape is awful complementary to this actin active site shape. There's going to be magic that happens here, y'all. This head is going to bind to this active site. It's going to be beautiful. Okay. Each of those myosin heads are going to bind with the actin active sites. Okay. So speaking of actin, it is one of our three thin filaments. Okay. Multiple actin subunits. Remember, each one of these little purple circles, okay, these little beads, each one of these is an actin. You can see we've got this double string arrangement, almost like a set of pearls folded on itself. Okay. These are going to be the ones that myosin is going to grab onto here at the active site. Okay, so these are going to play a direct role in muscle contraction. Okay. Troponin and tropomyosin are both regulatory proteins, so they are going to control when muscle contraction happens. Tropomyosin, the little blue rope, you will notice in this image, is covering up the actin active sites. Okay. In your head, if you can imagine a myosin head coming in here and trying to attach to any of these active sites, it would not be able to do it. This tropomyosin's in the way. Okay. That's exactly what its job is. Remember, we are regulating when contraction happens. When we are ready to do contraction, we are going to grab a hold of troponin and pull. Okay. Grabbing on and pulling on troponin, which is attached to the tropomyosin. Okay. Troponin will pull the tropomyosin out of the way. These active sites will now be exposed, so myosin will be able to grab on and complete muscle contraction. Okay. Okay, we know that skeletal muscle is striated. Okay. Let's talk about those stripes real quick. We have some bands. We've got light bands and we've got dark bands. Light bands contain only thin filaments. Dark bands contain thin and thick filaments, so we have more there, so we have a darker appearance. And this is kind of what it looks like. Okay. You can see we've got lighter areas, darker areas, lighter areas, darker areas. So let's make sure we know what's going on in each of these little sections. You have in your light and dark areas, okay, go back. Okay, so we have light areas and we have dark areas. Okay, light bands, dark bands. There are things happening, okay, within these regions. You have an I band, okay, I for light, thin filaments, okay, lots of I's. I bands are composed of thin filaments only, okay, these are in your light bands. In the middle, you have a Z disc. Okay, so if we go back to our picture, in the middle of this I band, this light band, we've got this dark line. This is your Z disc. Okay, this is your Z disc. If we kind of go up, 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 you'll notice that we've only got thin filaments here. We've got no thick filaments. Okay, these structural proteins in the middle here, okay, provide the Z disc with a little bit darker of an appearance. Okay, but these regions of thin filaments only, it's that lighter color. Okay. These structural proteins, including titin, remember these are going to anchor the thin filaments in place. They serve as attachment points for the elastic filaments, and they attach to the myofibrils, the myofibrils, excuse me, to one another across the diameter of the muscle fiber to kind of hold everybody in place. And again, we're also trying to prevent overstretching with these elastic filaments. Now, we had a light band, okay? Now I got a dark band, our A band, okay? This contains quite a few things. We've got quite a few things going on in our A band. It's kind of busy. Okay. There is a zone of overlap where you find both thick and thin filaments. Okay. Where you have this overlap, where thick and thin filaments overlap, is where you can generate tension and muscle contraction. Okay. If thick and thin filaments don't overlap, it's really hard to do contraction because thick filaments pull on thin filaments. In the middle of the A band, where you only find thick filaments, we call this the H zone. Okay. And in the very, very middle, we have the M line. Okay. This is again structural proteins that hold the thick filaments in place. And we are going to anchor more of your elastic filaments. So let's go back. Here we go. Here's our A band. Here's our dark band. Okay. Here's our zone of overlap, or thick and thin filaments. Okay, on both sides. In the middle is our H zone. This is only thick filaments. Okay. And right down the middle, we have more structural proteins creating the M line. Okay. Okay. All right. Just another image of the exact same thing. Just another cartoon picture. So we did talk I band, A band, Z disc, H zone, M line. We have thrown around all of those terms. Okay. And just to kind of add complexity, remember each myofibril with the thick and thin filaments compose your overall muscle fiber. Okay, so we've got tons of these myofibrils within your muscle fiber. Don't forget about your sarcoplasmic reticulum surrounding each myofibril as well. Okay, myofibrils have myofilaments in them. Okay, sliding filament model. All right, so if you take your hands, okay, like this picture. If these are our Z discs, okay, if our wrists are the Z discs, and our fingers are the thin and thick filaments, if you slide your fingers past each other, okay, that is representative of what happens during muscle contraction. Okay, so this whole configuration, Z disc to Z disc, that's a sarcomere. So, there's my picture. Here we go. This whole thing, Z disc to Z disc, is a sarcomere. Okay? Your myofibrils are sarcomere after sarcomere after sarcomere. Okay? Sarcomeres are the unit of muscle contraction. This is where muscle contraction happens. Okay? You will notice that your fingers do not magically become shorter. Okay? These are just moving closer together. They are sliding past each other. Okay? Again, that is exactly how your sarcomeres work. Okay? So again, your sarcomere, one Z disc to the next, functional unit. This is where contraction happens. Okay? So during contraction, the I band and the H zone are going to become more narrow. However, the A band will remain the same size. In order to make this happen, myosin heads are going to attach to the actin active sites. They're going to pull the thin filaments towards the M line, towards the middle, which brings the Z discs closer together. This shortens the overall sarcomere. Okay. We said that myofibrils had sarcomere after sarcomere after sarcomere. So with each myofibril contraction, we simultaneously contract all of them, really. This collectively shortens the whole muscle fiber. Okay, going the wrong way. Here we go. So relaxed, we've got I bands of a certain length. We've got H zone of a certain length. But contracted, these I bands are going to narrow. Okay, H zone is going to narrow. The A band, however, remember the A band is just the length of the thick filaments. And the thick filaments don't really move. It's the thin filaments that move. They get pulled inwards. So this A band does not change. Okay? But remember, we have sarcomere after sarcomere. So when this contracts and starts to pull, the next one contracts and pulls. So collectively, your muscle gets quote unquote shorter during contraction.